roadmap. Thanks. Here's a little roadmap for this talk to give an illusion of linearity as someone who likes to bounce around. I'll talk a little on hallucinogenic states and how we might start to frame them in the context of brain activity. And I hope to get you intrigued philosophically and empirically about this question before trying to convince you where to look when trying to study altered states, at least again in neuroscience. And it'll be biased by my history, of course, and my intrigue with and love for the memory area, hippocampus, and its collaboration with sensory areas. I'll continue by describing the 5-HT2A receptor and its role in altered states, and also in healing, which you've heard a good deal about today. I'll eventually get into some of my work describing the methods we use and how memory and visual areas of the brain work together normally, and then how that changes with LSD. We can speculate on the parallels between the firing changes in brain cells recorded and also the reports of experience in humans. So we'll bring it all together to talk about the investigative approach that we need in psychedelic science. And excuse some tangents or explanations along the way, but I really want to impart understanding of these approaches in the field because understanding psychedelics is, as Martin described, a wicked problem. I learned that term today. And wicked problems are very hard to solve without interdisciplinary crosstalk. So most importantly, I want to reinforce what you've seen today, hoping that you see those parallels and also seeing how my data aligns, despite them all being very different techniques used to study. Broadly, I want you to learn some things, ponder some things, and take away why and especially how to approach studying these hallucinogenic states. Not how and why I study necessarily, but broadly leave you seeing why intersection among disciplines in the sciences is crucial. So let's start talking about altered perceptual states. We can think specifically on hallucinations. A hallucination experience is when an individual reports that a percept is incompatible with the world around them. This can be a visual distortion or an auditory experience, for example, but the manifestation of that experience does not align with the external world inputs, at least not how the average human is experiencing it. Obviously, there's a lot of philosophical pivots on the topic, but what I want you to be considering is at the neuronal level, in the brain, what might be happening in this case. Hallucinogenic psychedelic drugs like lysergic acid diethylamide or LSD confer hallucinations, but they are also experienced, those hallucinations are also experienced in pathologies like schizophrenia and Parkinson's disease, among others. So generally, waking perceptual experience manifests in a brain cell response to a physical input. For example, a sound wave impinges on auditory hair cells in the ear, which is electrically transmitted through a pathway that fires up neurons in the auditory cortex and so forth downstream. I could record from your auditory cortex when I play certain sounds, and I could predictably see neurons organized in a certain frequency map fire when I play those specific sounds. So when one experiences auditory sounds that are not from a sound wave, you can imagine various hypotheses. Is auditory cortex going rogue, firing from its own aberrant triggers, or is it coming from a higher level brain structure, pressing the auditory cortex's buttons, sort of like maybe when we're dreaming? Is another brain region no longer suppressing the auditory area, allowing it to kind of party on without supervision? I just want you to be considering these questions going forth in this talk, but also in your journey learning more about psychedelics, again, especially those that induce these types of altered perceptual states. And I also want to instate maybe a degree of consternation and perplexity, recognizing how difficult it is to study what's happening in the brain with these drugs. I emphasize altered percepts or hallucinations because this generally occurs with serotonergic drugs that agonize and activate the 5-HT2A receptor. So you've heard again about this receptor quite a bit today. It's critical for the hallucination experience. If you block this receptor with another drug or an antipsychotic, psychedelics like LSD or psilocybin are far less effective at inducing sensory distortions. Blocking that receptor is in fact used in patients that experience visual or auditory hallucinations in those pathologies that I mentioned. In my data today, I'll be showing you neural activity responses to LSD, but also to LSD with a 5-HT2A blocker condition combined. While up for debate, it seems as though this receptor also confers a great deal of therapeutic relief. Talks today mentioned plasticity as being part of that puzzle. In some studies with that receptor blocked, patients with major depressive disorder did not report as substantial therapeutic benefit as they did without it blocked, when they were given psychedelics like psilocybin. I also like to mention, as Reagan did earlier in her talk, actually just really recently, on her talk in Language and Consciousness, and Gerardo was talking about ayahuasca, that participants given these drugs generally report psychedelic hallucinogens. Those participants generally report feelings of interconnectedness, ego dissolution, even boundlessness, which those sentiments are also blocked if that receptor is blocked. So these are fascinating and complicated sentiments associated with psychedelic hallucinogens. Ego death, I can't really tell you how that's computed by specific brain cell responses. I'm going to leave network dynamics that confer the experience of my concept of self dissolving to the computational modeling folks for now, like Martin. My lab does study other neural encoding, however. We see how specific signals are encoded and passed between brain regions to create a cognitive map of the world. How sensory signals, like visual cues or auditory sounds, passed on and integrate to update an individual where it is in space. We also study how these mental maps support memories later on. We look at firing activities like these to investigate memory. Episodic memory binds together tons of different data content and it anchors it to space and time. A hub for this type of integration of info is in the hippocampus. So right now, your hippocampus is taking inputs from your sensory areas. In wake, you want to encode this memory, but you also need to know where you are in the world. Boom, hippocampus. The map lights up and it tells you that you're in your house and it filters incoming information. Higher order areas filter out the air conditioning and amplify my voice if you're paying attention to it. So those sounds exist, but they're gated, right? And what's important gets incorporated into memories down the road. In sleep, you get consolidation and the information flow sort of reverses direction. You have the reactivation from places like hippocampus, exciting and activating the sensory regions in that direction. So I'm going to spit a few more facts at you, but they are all related. They're all why hippocampus is this fundamental place, in my opinion, to be looking to understand both typical consciousness as well as to understand hallucinations and healing. It's a major driver when you hallucinate every night in dreams, in which sensory activation does not come from incoming external inputs. The hippocampus also is hyperexcited and eventually atrophied in schizophrenia patients that have auditory and visual hallucinations. The fiber tract that connects these sensory cortices to the hippocampus are morphologically different depending on a symptom that the patient has. So if they have visual hallucinations, the tract between visual areas and that temporal region of the hippocampus is altered, for example. And if you stimulate a human's medial temporal lobe, right where that hippocampus is housed, that participant will experience visual hallucinations. A final plug I'll say about hippocampus, I've suggested but not quite stated, is that it is the seat of episodic, otherwise known as autobiographical memory in humans. Memories that self-reference, memories associated with both joy and also of sadness or guilt. Echoing some of what you've heard today, like Gerardo talking about self-referential networks, which quiet down with meditation and ayahuasca, autobiographical memory networks, including the hippocampus, are overactive in patients with major depressive disorder, patients that often struggle with ruminations, repeatedly remembering negative memories or that have negative self-talk. These are also the same patients that the data is showing benefit from psychedelic hallucinogens. This is a major place to be looking and to understand the therapeutic potential of 5-HT2A agonizing psychedelics, which we already know demonstrates some benefits, but we need a careful, well-investigated approach to carry on. So yes, hippocampus is a target of interest, obviously, especially in my work. We have a pretty good idea of how it operates to typically create a map of the world that I mentioned, and visual cortex does as well. We know these two regions coordinate constantly to update your present experience. I can't, however, record from these regions and listen to the neurons talk in humans. So in our lab, we record from the brains of rats. We do so in a way similar to Zarmin Zahid said earlier in her work. We implant devices in the brain surgically, but in this case, we're actually implanting tetrodes, which are electrode bundles, into our regions of interest. Instead of stimulating and recording downstream, as in the other talk, we are just letting the cells do their thing, and we're recording the extracellular activity to see what the cells are communicating to one another. What are they encoding about the world? This is too invasive for humans right now, except for some epilepsy patients, but it gives us a wealth of data when done in an animal model. What are individual neurons responding to about an experience of the rat? And also, how is the whole population responding as a whole? If you saw Gabriela Sawicka's talk earlier on brain dynamics, she described some population level recordings in humans. It gives insights into brain states, which she called the harmonic mode of the human connectome or network. Population code, or the oscillatory activity of many, many neurons at once, allows for individual brain cells to participate in the symphony. That symphony is not only important within the brain region, like in hippocampus for an individual cell to be bounded tightly with another cell in a current map, but it's also important across brain regions. Powerful frequencies or rhythms have functions to deliver their message properly and coherently to other parts of the brain. It's nice because in rats, we can examine the symphony if recording from more than one spot at a time. And we can see if the symphony changes its organization with psychedelics. We can see the discordance or the coordination, and we can make an informed guess about what that might mean about conscious perception or altered perception. So different states involve different networks, right? We've heard that a lot today. When I'm sleeping in memory consolidation mode, I've got certain parts of the brain online, and the direction of input is one way. I mentioned temporal areas like hippocampus feed towards primary sensory areas when they're working to bind together information from the day. You don't have stuff quite as gated or as modulated, right? Like in dreams, unexpected stuff happens. You don't have inhibition from higher order areas, quieting possible errors, allowing for some bizarreness, essentially. You might consider that in dreams, experience is less constrained. When I go on a run, that's a different state entirely. You've got your motor loops, navigation circuitry, sensory inputs, they're all working together, all coming in a different direction. So we recorded from rats in different states in the study that most of the studies that I've done. And we look at their laps whenever they're running for a food reward, which is considered like an active state, as well as during stops, as well as during sleep. So they have periods of immobility, and they also have sleep sessions that we're kind of trying to compare these different states across from. And rats were pre-trained to run laps back and forth on the C-shaped track that you see. And we recorded from neurons in two parts of the brain that you can probably infer now, the visual cortex, mostly primary visual cortex called V1 throughout the talk. And the other region is CA1 of hippocampus. So Armin talked a little bit earlier about ventral CA1 of hippocampus, and we're recording in the dorsal part, which is close by, but has a different connectivity and also different functions with a lot of place cells. And we'll get into that in a few. We had our rats run in a pre-session with a saline control drug, and then they slept, and then they run a post-session. So this allows kind of for within subject control and then also between, as our post-session can either have a control drug or it will have a very high or low dose of LSD. Our control condition, I briefly mentioned earlier, includes several different conditions, including a saline injection, the 5-HT2A receptor antagonist known as M100-907, or that blocker plus LSD. And for the sake of my results, I show all the controls together because they were indistinguishable from one another in the results that I show you and in their behavior, which is very interesting. So for our question, we know quite well how this region of the hippocampus encodes a map of the world. Imagine you have a map of your driveway. Your place cells represent each section of space, kind of like the squares here, and they come together to form this map. They fire depending where you are. If you're standing here, one's firing right there, and all along your walk to the mailbox, you'll have these place cells firing alongside regions of the brain, other regions of the brain that are relevant to that spot, sort of wrapping it up into a little episodic memory burrito. The visual cue of that mailbox is tightly bound not only to your full map of the driveway, but the spots beside the mailbox. So see how the section of the map where the mailbox is tied to the visual cue of the mailbox. So visual neurons encoding the mailbox are partnered up with place cells, and these regions they're not the same region. They're different parts of the brain coordinating together to construct this map of reality, working together. So this is a really nice way to consider how the brain encodes and stores experiences in a sort of, smaller way, but very clear to see these relationships. And you got your place cell firing there. So now let's look at that basically exact same thing again, but in the rat brain. Here we have a linearized track, so that C-shaped track is now just put on a straight line so that you can visualize easier like what's going on. And position is on the x-axis. And the rat here runs from the left food well to the right food well, and those laps are stacked on top of each other. You can see that the CA1 Place cell fires reliably in the same spot. The raster is the neuron firing off an action potential in that location, sort of by the corner of the this at least corner of the track every single time. The curve at the bottom is the mean response curve of the neurons in space across all the laps. It's a really nice way to see like the place cell tuning across the whole trajectory. So you can imagine you get a really nice map, kind of like what's depicted on the bottom, if you put all the place cells together of the whole track. You can know where the rat is in space just by hearing what neuron is firing. So we see some visual neurons that also prefer that corner. So plotted on the right here is a red colored visual neuron from V1 on the same exact laps, same rat. And you can see how that cell fires at the two corners, sort of like the mailbox. Those two cells in hippocampus and visual cortex fire in two different brain regions, but close to one another in time and space. So cool stuff about neuroplasticity, fire together, wire together. Through memory consolidation, they become coactive, not just during the experience, but later on during rest as well. So this visual neuron in red likes the corners. It fires at both corners because they look the same, and visual neurons encode visual stimuli, right? But so what else happens in the brain, right, is that systems work together to filter and refine, like we talked about filtering out the AC, or whenever you're attending to something. So over time with experience, or with more training on the track, the visual neuron becomes so tied to this place cell that it preferentially fires at that location, at this first corner, and not as much at the other, despite looking similar. It is tightly bound with the memory region by both increasing and decreasing in firing rate with one another. If you look at specific laps on the right, you can see that both brain cells from different areas co-fluctuate together. When one increases, so too does the other. They are tightly bound by their experience over time. As you take more walks around your neighborhood, or as the rat runs more laps in the track, one corner becomes tightly bound to a location in space, to place cell firing in hippocampus. So this is a result of modulation, another word that's been mentioned a bit today. It helps conserve energy in the brain, and it also helps you get better at using information and becoming a great prediction machine. Walking outbound from your house has a certain map tied to certain cues and memories. Happy phone calls associated with a specific map, for example. So this is the firm beauty, the blessing, and the curse of episodic memory. Patterns get ingrained and they get more efficient. It becomes easier to activate learned memory. This rule makes humans efficient for operating in the world, but can also debilitate when it goes awry. When autobiographical memory, strongly bound to negative emotions, is strengthened and perseverated on, as occurs in things like depression or PTSD. So that's food for thought going forward. Perhaps hallucinogens like LSD can loosen up this relationship to quiet priors coming from top-down areas like hippocampus. If you loosen those priors, you may get less gated sensory areas, and you also may get less, a loosened grasp of self-referential autobiographical processes. This isn't a novel suggestion, but these are novel results and data and new experiment parsing that can sort of support or deny these types of hypotheses. So this tightly bound relationship makes operating as an organism efficient, but it also constrains experience. Let's quantify this relationship, this partnership we see. So to quantify the co-fluctuation and the concordant dialogue between the visual neurons and the place cells, we can take basically this is my little data section, and hopefully it's graspable. We can take all of our possible active cell pairs between the two regions, and we'll see a cross-correllogram of their spiking activity during track running. So first, we normalize each cell's firing for this type of computation because generally visual cortex fires more overall anyway. They just tend to behave a little bit differently. So to see the relationship better, you normalize first, and then you can correlate the two at different time lags, starting at time 0. So if two things are coincident, you'd expect them to be highly correlated if they fire at the same time. So in this example, which you've seen a couple of times now, you'd expect a pretty high correlation value with 0 time lag, since they look like these 2 cells in these 2 different regions fired about the same time consistently. And we do see just that. These values displayed, they subtract the same result applied to cell pairs with a shuffled firing across time bins. It's just a typical way to kind of analyze these things as a control. If they are commonly coactive, firing coincidentally opposed to random firing, that again value will be closer to 1, and that's what we found, as has been demonstrated before, between these 2 regions in a typical rat. So that's a significant co-firing or coactivity correlation between these 2 brain regions without LSD, right? So now let's get to the LSD part. If we give a high dose of LSD, which is known to, again, induce a hallucinogenic altered state, what happens to this relationship? The relationship alters. So the post on the right is post-LSD. The pre is the pre-running session before they were administered the drug. So that relationship alters, and the 2 regions crosstalk weakens. That co-fluctuation that we saw subsides, and that deeply established relationship between the cell pairs in the visual area in CA1 decouples. So the change in the relationship is further explored in our lab's paper that was published last year that was mentioned. And I can tell you that this coactivity continues to be altered in other states, like rest. But we'll move along in the interest of time and to keep this talk as accessible as possible. And again, I want you to be thinking, though, of the parallels of these findings with the talks you heard today. Much was shared on changes in brain states and connectivity at the global scale in humans. And now we're zooming in a bit to see the actual neuronal chatter. How is neuronal activity overall changing in this memory and also the visual sensory area now that they are decoupled? Within their respective regions, we can now look at their overall mean firing rate comparisons during running. So how much did all the neurons fire after controlling for behavioral confounds? We have to control for confounds like speed first, and then we can kind of see what the mean firing rate is like, the main activity. Did CA1 activate more or less in this active running state with LSD? We saw that when the relationship was strong, if one fired a lot, so too did the other. So a little more data here. Now you're looking at a cumulative probability distribution of that mean firing rate for active cells. So all cells are active in both pre and post. So that our effect is not a matter of just them becoming silent. And so, yeah, you're looking at those active cells before and after LSD, otherwise pre and post, although the control is pre and post saline, or pre and post that blocker that I mentioned. The red will be our LSD example. So CA1 active cells fired about the same rate during running in both sessions and controls as you would expect. But you can see that post-LSD, the firing rate dropped substantially in the hippocampal CA1. So that thick red line is showing you that essentially the mean rate is much, much lower in the post session. The thicker lines are the post sessions across all of these plots. And then in visual cortex, the overall activity stayed about the same. So now again, LSD lowers hippocampal activity in this active state, while visual cortex is just about as active as before. We saw what was going on with those CA1 Place cells that stopped firing as much. And I'll rather quickly tell you that while their rates are lower, the map remains. Oh, can you see that? Cool. The map does remain. So it's not like you now have this whole new map or an aberrant map or something. Spatial information is intact. Hippocampus does recognize that the rat is in the corner, for example, from its earlier experiences. So if place cells are still correct, visual neurons did not follow their activity as usual. Visual neurons kept active rather than staying bound to their ingrained modulated circuit, in which they would, typically fluctuate again with those cells in hippocampus, decreasing or increasing with the place cells. So, and then I will mention again, we showed plots like this earlier, but this is another place cell tuning of another example cell in pre and then in post. And you can see that it's spatial tuning, or spatial information is still tuned to the same region of the environment. The rat in the middle of the track here, the place cell fires. And that's pretty much the same as well in the LSD condition. So that map's not changing. The firing rate changed with LSD in lab running mode. So in hippocampus, the place cells fired substantially less frequently. But visual areas continue to fire as often as typical. And we know again that they're not coactive at this point. So if CA1 is reduced in firing, we can posit that its modulation perhaps, or its constraints on visual cortex have likely been loosened in this state that is generally associated with altered perception. So interestingly, CA1 is also less constrained. A result that I have to mention, but didn't want to show too many data figures, so I'm just going to mention it, is the following. Please see my paper, email me, or ask me for more details. But within hippocampus itself, we also found that the place cell behavior changes its response to the environment. So those place cells actually, through some other ways of measuring them and seeing how they act and respond to the world around them, they act like they do earlier in training, essentially. So the first few days of training, when a rat is first establishing a map and memories of a place, those place cells usually fire less discriminately, sort of like how our visual neurons first fire at both corners. With more experience and more priors, more context, they become more specific. And we found while in LSD that those place cells acted as they did earlier as well. They fire as they did when the environment was newer, and again, the experience was earlier. In our experiment, they fire regardless of trajectory with LSD. So they are trajectory specific and controls. The trajectory rigid encoding loosens with LSD. So this is another mechanism that could be attributed to the plasticity that goes hand in hand with the benefits of these drugs. Perhaps a loosened window to allow unlearning or relearning. Again, this loosening cognitively may echo the perceptual experience of reduced constraints from other brain regions, all things in future directions to be further parsed. So I'm going to show you one final result to consider in the context of what could be going on in these hallucinogenic states. Let's return to the fact now from the previous slide that our hippocampal neurons did quiet overall with LSD in an active state. So CA1 neurons and hippocampus, our episodic memory hub, get quieter across the board. That means not just in running, even in memory activation during immobility and rest, a different state, we saw that they got quieter. So bursting firing events in the hippocampus called sharp wave ripples are a signature of memory replay events. I love showing this. I was gonna not show sharp wave ripples because it can be a little bit confusing, but they're just so cool that you have to kind of see how memory can be studied and how it's seen in the brain. So there are these beautiful and powerful bursts of memory code that facilitate, again, strengthening memories, and they occur during memory recall as well. So in the EEG recording, it looks like that fast, high amplitude event, that big ripple that's right there to the right. And the neurons in the map fire in the order of the experience within it. So on the left, you're kind of seeing the color of, the cells firing temporally as the rat would walk across. And you see those bursts very rapidly within these sharp wave ripple events. Another example of the global orchestra in the brain. So when a rat thinks about a fearful memory in which it was shocked, for example, in an arena, the map where he walked when he got shocked lights up and it fires in a sharp wave ripple replay event. You even see that before he turns and avoids the region. So when you think about a traumatic experience, for example, your map bursts, not only with the spatial code in CA1 of hippocampus, but it's actually so powerful that it recruits the rest of the pattern, the limbic or emotional part of the brain, the visual areas associated with it, those visual cues. All of that context lights up with that sharp wave ripple. But with LSD, we see that those ripples weaken and they don't even occur as often in the LSD state in our experiment during immobility and during rest. So this is a consideration, again, for future study, a possible mechanism for therapeutic relief if it can be harnessed together with proper therapy. To draw parallels to human research, these ripple events occur when a participant thinks about an episodic memory, like I had mentioned, a process that is out of control in cases like depression. This finding also shows the reduction of that top-down grasp. Again, that's occurring in the psychedelic state. Less constraints from hippocampus on bottom-up processes as a consequence of network dysregulation, like we again saw earlier today. So I only showed a little bit of data now, but I hope that you've learned some interesting stuff about this, that you've seen how there's so many different techniques, so many different ways to study, so many brain regions and big players throughout the whole day, and then they also included in this study and whatnot that can help us inform and kind of pave a way to try to understand what's going on here. Episodic memory requires global coordination of the brain. It requires the symphony, again, of brain regions to couple together, to encode as you go about your day, to reactivate when you recall a memory, and to strengthen and consolidate during rest. These pathways are strong, they make brain functioning efficient, but they can also be co-opted to manifest in suffering, like if a potent negative memory becomes too strong and revisited. The plasticity-inducing effects of psychedelics like LSD seems to be critical to help loosen the reins, to make a circuit more flexible. You must quiet the exertion from the top-down mechanisms. Higher order areas of the brain, like hippocampus or prefrontal cortex, need to be quieted to let expected or ingrained processes quiet. Visual distortions associated with LSD are reportedly less bounded, less gated. Perhaps the sensory boundlessness parallels the complex cognitive experience of boundlessness. Perhaps the visual experience echoes the cognitive. Cognitive memories, traumas, rules get loosened for that window of time. We saw that place cells loosen their grasp on visual responses with LSD. We see that the memory area exerts less control and even reverts into earlier stages of experience on an operational level. In summary, my results show insights relating to neural signaling during a hallucination state. Remember that with LSD, patients are deriving healing from one use in a clinical setting. These patients have pathologies that involve deeply ingrained thought habits. And another consequence of LSD, again, is that it alters that sensory perception. So I know this has been a ride darting. Oh, I keep forgetting my one liner of that implication, essentially. So if LSD loosens and opens circuitry that's well established in these thought disorders, perhaps the hallucination is the sensory manifestation of that same loosening. Less bounded activity that was again shaped initially by prior experiences and expectations. And yeah, this has been really fun to talk about, and I had to get a little bit in when it comes to this stuff going on in the place cell field, especially because there's very, very few, if any, like there was, I think, one study that looked at place cells with DOI whenever I defended. So this is another new place, a new avenue to be studying this kind of stuff and looking at things like memory and plasticity. So that's why I had to illustrate these angles, these different angles that we have to take in the field. We had Gabriella talk about modeling and large-scale dynamics with psychedelics. So Armin talked about the circuit level with receptor expression and postsynaptic currents to look at plasticity dynamics. We have huge repositories of anecdote on the internet and patient data reports. We need to continue to attack these questions from all directions. And we need more answers to how these drugs exert their effects before we can move forward in implementing them across the board. Unlike rodents, humans have a great deal of complexity, including language, which Reggie discussed. Language invokes its own constellations of brain regions. She said that thinking of garlic activates your olfactory bulb, memory circuitry, and more. So to get solid healing, we have to continue to compare the global fMRI recordings, like her study, with the neuronal dialogue that's happening in rodent studies. We need to inform computational models like Gabriella described and cross-check that with patient reports. And of course, though, like Uma talked about, and I'm sure we'll hear a lot tomorrow as well, we have to be careful. We're scratching the surface right now, but we don't know what conditions could actually be exacerbated by psychedelics. Like how Uma showed MDMA therapy can actually be harmful for patients with both OCD and PTSD, and how Javier discussed comorbidities between ADHD and other pathologies. Healing, especially mental healing, is not one size fits all, as has been tried and failed with current pharmaceuticals. Martin discussed further how modeling can help alleviate and predict problems like this, but modeling requires biological data to inform. These are ways to treat the patient, not the disease. As Javier said, we need to treat the patient, not the disease. I hope you see how in the place cell field, there are findings that inform us to more and more questions and thus more and more answers. And then of course, more and more questions. I brought up 2 critical regions of the brain for constructing reality. And we probed it to get an idea of what happens when that reality gets distorted, how they communicate differently, and especially encode external reality differently. It leaves many more questions, but sharing with you and others helps the field progress more quickly. Thanks for being part of that multidisciplinary conversation. I'd like to thank my mentor, Daoyun Ji, and also the contribution from all of the rats in my studies. And I appreciate you for your attention very much. Here are some of my references, not quite all of them, so if it's not on here, I can email you anything you might be interested in. Yeah, so thank you.
Awesome. Thanks so much, Carli.